When plant breeders cross two elite parental lines, they create an F1 hybrid. This first generation is uniform, but it sets the stage for the F2 generation, the offspring of that initial hybrid. It is in this F2 generation where massive genetic segregation occurs, producing a sprawling population where every single plant is genetically unique. This chart shows the strict mathematical decay of heterozygosity, the state of having different alleles or gene variants for a specific trait. In self-pollinated crops like wheat or soybeans, each subsequent generation naturally loses 50% of its heterozygosity. The population is rapidly driven towards homozygosity, meaning the genetic traits become permanently fixed and uniform. The plant breeder's primary objective is to evaluate this chaotic, segregating population and isolate the absolute best genetic combination to form a pure line cultivar. They must accomplish this before the genetics become permanently fixed around the F6 generation. Because of this rapid biological decay, the ultimate challenge in plant breeding is logistical. It requires engineering the physical operations necessary to grow, evaluate, and select from thousands of segregating plants within a constantly closing window of time. To manage these segregating populations, geneticists rely on three primary operational frameworks: the pedigree method, the bulk method, and the selected bulk method. Choosing between these three approaches requires a strict negotiation. A program must balance the precision of its genetic selection against the expenditure of its resources, namely land, labor, and data management. This creates a constant tension between throughput, the physical ability to evaluate millions of plants in the field, and tracking, the administrative ability to know the exact genetic parentage of any single plant. Maximizing one of these variables inherently limits the other. If a breeder tracks every single lineage, they can only plant a restricted number of crosses. If they plant massive populations, they cannot physically track it all. Breeding programs adopt a specific logistical architecture based on their available funding and scientific targets. The chosen operational architecture dictates exactly which genetic traits can be successfully captured and which will be lost to the noise of the field. The pedigree method is the most data-intensive approach available. In this system, the precise genealogy of every selected plant is meticulously recorded across every generation. This diagram outlines the pedigree framework. In F2, breeders select superior plants. In F3 and F4, seeds grow in dedicated progeny rows to evaluate heritable traits. Tracking lineages requires complex alphanumeric tagging to connect an F5 plant back to its exact F2 ancestor. The major advantage of this method is control. It allows breeders to call visible genetic defects extremely early and accurately trace specific allele inheritance over time. But the pedigree method has a critical bottleneck. As families multiply, the required record-keeping and field plots explode exponentially. This administrative and spatial bloat physically caps the total number of crosses a breeding program can realistically evaluate in a single year. While the pedigree method offers unmatched genetic resolution, its labor-intensive nature makes it inefficient for discovering complex multi-gene traits like overall yield, which require massive population sizes to isolate. The pure bulk method is the direct logistical inverse of pedigree. It prioritizes massive population sizes by completely abandoning all lineage tracking. This workflow demonstrates the bulk strategy. From F2 through F5, all plants are harvested together. Seeds are mixed in bulk, and a random sample is planted the next year. Notice the volume of seeds. During these generations, artificial selection is entirely suspended. The biological strategy here is to subject the population to intense natural selection pressures from the environment, such as drought, disease, and competition for sunlight. Because no individual tagging is required, this method is highly cost-effective. It requires minimal labor, allowing breeders to handle vastly larger populations across significantly less land. However, pure bulk has a distinct flaw for commercial agriculture. Natural selection favors aggressive competitors, plants that can outshade their neighbors. It does not necessarily favor plants with the highest agronomic yield when grown in uniform commercial stands. By the time artificial selection finally occurs at the F6 generation, when the lines are mostly homozygous, rare but highly productive genotypes may have already been choked out by taller, more aggressive variants. Relying entirely on natural selection is an excellent tool for building evolutionary hardiness, but but it is an inefficient framework for systematically engineering high-yielding, market-ready cultivars. To fix the administrative bottlenecks of Pedigree and the yield and precision of pure bulk, global institutions like CIMMYT engineered a hybrid solution known as the selected bulk method. In the F2 generation, breeders visually evaluate and select the absolute best individual plants based on agronomic traits, exactly as in the Pedigree method. But here is the critical divergence. Instead of keeping seeds separate to track lineage, seeds from all selected F2 plants are immediately pooled into a single premium bulk. By discarding this lineage data, the program eliminates the exponential labor cost that cripples the Pedigree method. This curated bulk is then planted in the F3 generation and exposed to artificial disease epidemics. Only the best surviving plants are selected, and their seeds are bulked again. Genetically, this aggressively enriches the population's allele frequency for desirable quantitative traits early in the cycle, without sacrificing overall genetic diversity. Because field labor is entirely decoupled from record-keeping, global breeding programs can evaluate vastly more crosses in the exact same amount of physical space. By deliberately choosing not to track data, modern programs unlocked the logistical scaling required to breed complex quantitative traits like yield across global environments. Modern breeders deploy these three distinct architectures strategically, based entirely on their specific genetic targets. The Pedigree method remains the gold standard when a breeder must track the exact inheritance of highly heritable traits, or when characterizing completely novel genetic sources. The pure bulk method is deployed primarily when a program needs to adapt a large population to a specific, harsh environmental stress over many years, usually with minimal funding. Selected bulk dominates modern high-throughput commercial breeding programs tasked with maximizing general yield and broad adaptability. This image illustrates that scale. Selected bulk allows programs to evaluate thousands of advanced lines simultaneously, a physical operation mathematically impossible under strict pedigree systems. There is no single perfect breeding method. The operational architecture must be strictly dictated by the biological objective of the program. The inescapable ticking clock of heterozygosity decay in self-pollinated crops forces every decision a breeder makes. Overcoming this constraint requires mastering supply chain logistics, probability, and resource allocation. In the science of high-throughput plant breeding, deciding what data to safely discard is just as critical as deciding what phenotypes to select. Ultimately, true genetic gain is achieved only when deep biological insight is paired with flawless, ruthless operational execution.